So David Jarrard, I'm a physician here at the University of Wisconsin Comprehensive Cancer Center. My role is both clinical, I take care of patients, but we also have a research lab.
Oh, awesome. So what do you do in your research lab?
So there's two main questions that we study, and one of those is looking at the incredibly high incidence of prostate cancer in men as they age. And our lab has discovered that there are epigenetic reasons for why cancer arises so commonly in men as they get older. The second area that we're interested in is reinducing an arrest in cells, cancer cells, and we term this senescence. So we're interested in studying new drugs or chemicals that can cause senescence in cancer cells. So we're not so much interested in killing them as we are stopping them from growing.
So why don't you tell us how epigenetics plays a role in your lab specifically?
So with regard to many cancers, it was clear that genetic changes only partially explained what was happening in these cancers. And the field of epigenetics is interesting. Again, as you all know, epigenetics are essentially protein changes on the DNA that alter its function. And the neat thing about epigenetics is that it can actually be reversible. So when one thinks of epigenetic changes in cancer and cancer development, there are ways of actually reversing these changes. And one clear way that our lab functions is always asking the question, how can this help people? So one major area of interest is how you can reverse these epigenetic changes to prevent cancer or reverse the cancer phenotype.
Yeah, so basically with cell lines, the cell lines that we use are human prostate cancer cell lines. And in cancer research, kind of a common way to do cancer research is, if you change the expression of a certain gene in a cancer cell line or more of a normal cell line, what happens to the cells? Do they grow more aggressive? Do they slow down? And that's kind of the classical way that people kind of figured out what genes were cancer related or important to cancer progression or development. So what I'm doing right now is I have some cell lines where I've actually altered some specific genes of interest that we're interested in, and right now I'm just kind of passaging and keeping my lines going and setting them up for some other things, but the main idea behind it is we knock down a gene and we try to see what types of cellular changes happen. What happens with other genes or other cancer-related genes in these cells.
So what does a typical day in the lab look like to you, and what kind of stuff do you use in the lab?
So we spend, there's some members of my lab who spend all the time in the lab. I just spend part-time in the lab. Part of my duties are clinical. But this is a very common instrument we use in the lab. This is called a PCR machine, or polymerase chain reaction. It allows us to take small amounts of DNA, either native DNA or altered in some way, and amplify it up so we can detect it and manipulate it. So it's a very common thing that most labs utilize at this point. The person who first invented the enzymes involved in polymerase chain reaction actually won the Nobel Prize.
So where do you see the future research in your lab heading?
So the area of epigenetics is one that is really changing significantly. And as we uncover, it's almost like an onion, as we uncover each layer, there's another layer below that. But where our lab, everything we ask tends to refer back to what's going on in the clinic. For example, we're using some of the data we've gotten to look at changes in epigenetics to predict cancer outcomes and also identify individuals that have cancer. So epigenetic processes can be used as what we call biomarkers. As I mentioned, also seeing if you can revert these is a very important component of the future where this lab is going. And then finally, coming up with epigenetic modifiers, drugs that may kill cancer cells via attacking these altered epigenetic processes is again another important component of what we're working on.